everyone. Hi, how are you? This is a good, nice Saturday afternoon. Okay, how's everyone? Okay, so I'm sure that everyone must be relieved that you already finished one paper of your biology, which is paper 3, the Amali. Okay, the practical session, and you're all ready to face the SPM next week. Okay, so next week it's going to be a very busy week, all right, and the next week after that it's going to be even busier week when you have all 4 papers. There are 4 days of exam, so next week is only about, I think, 2 days of exam. Okay, so I see who, let me see who is in the channel with me today. Who is in the, good afternoon, RFLC Dutch. Okay, hi everyone, hi. Joanne, good afternoon. Jiawei, good afternoon. Min Yin, okay, good evening. Evening, afternoon, okay, same thing lah. All right, this is not night yet, okay. So good evening, everyone. Okay, good afternoon. Okay, today I'm going to tackle a topic which is one of my favorite topics actually, and it's not difficult actually. A lot of people are afraid to answer this question. Okay, once you know the concept and your concept is very sturdy, I mean you're very sure about your facts, you can answer questions. Actually, you can answer questions on this topic, inheritance and variation. So I noticed that this question is quite popular, right? Quite popular, although I would not promise that it will come out every year, all right, but it's quite popular. And if it's not related to that, sometimes it's related to your cell division. It could be a part of the question for cell division. Okay, it can also be a part of the question for, let's say, reproduction. So they, they can always mix in one question. Okay, so ex potato, hi, good evening. Right, everyone, let's get ready. Yeah. So I'm going to share screen now. I hope you can see it. 1 minute. All right. Okay. So this is my topic here is variation and inheritance. Okay, because I noticed that when I look at the questions for inheritance, there are also certain parts of variation that they throw in. So I cannot separate them. So, and sometimes it has to do with genetic engineering as well. So all these are interrelated. Okay. Jadi bahagian ini adalah what do you call that? Berkaitan dan bersambungan. Okay. So in my slide, again, I have 2 bahasa. All right. Mostly I will speak in English, but those who are taking it in BM, you can also see the answer there. So I'm like, catering to everybody. All right. Let's start now. All right. Let me see. All right. Okay. Now, let me start with a question, which is one of the questions that came out for the trial for one of the negri lah, right? One of the negri, the states. Okay. So this question is on inheritance of the comb shape in chickens. Okay, so diagram 8 shows the inheritance of 2 comb shape in chickens between a heterozygous buttercup comb chicken. So the buttercup is the one which is, the one that is a little bit bigger and it's like, it stands up. And then the homozygous strawberry comb chicken is a bit smaller. So the keywords here to see are, okay, here, don't forget which is dominant, which is recessive. So you need to have some knowledge about this, because I'm not explaining from beginning. I assume that everyone is already have the knowledge, you already revised, and you're just learning the technique to answer the questions. Okay, look at the keywords here. Heterozygous, they already mentioned to you that buttercup comb chicken is heterozygous. Okay, that means it has a dominant allele and a recessive allele. And then look at this one, homozygous. Strawberry comb chicken is homozygous. That means they have both the same alleles. 2 of them are the same alleles. Okay, if you look at the question, state the genotypes of chicken P and R. Okay, nyatakan genotype P dan R. Okay, so look here, genotype here you need to write in symbols. That means the BB, whether it's big B, small b, or small b. Remember, okay, you're not saying the phenotype. Phenotype is just the word itself, like strawberry comb or buttercup comb. That is your phenotype. Okay, so you can see here very clearly that heterozygous buttercup, this is your P. Okay, you see here, this is your P. It already tells you that it's heterozygous. So heterozygous means we have 1 dominant and 1 recessive. Whereas the other one, R, you can see it's exactly the same as Q. Your R is the same as Q, which is already mentioned is homozygous. So because it's strawberry, is must be the other type, so it has to be the recessive one. Okay, so the answer here is B, big B, small b, right, for the P. Whereas for the strawberry, because it is not buttercup, it is the other one. So it must have, this is the recessive trait. Recessive trait means it must have 2 recessive alleles. So small b, small b. Okay, so the first question is actually very easy. You already got two marks in your pocket. All right. Okay, let's go on to the next. Where you are, expert, I know where you live now. Okay, because I know this is a degree paper. Okay, I hope you score very well for it, exportator. All right, if not, then here are the answers. Okay, now draw a schematic diagram. I noticed that many students can draw, but the difficulty comes when they want to explain why they get this offspring. Okay, cara nak lukis tak susah, boleh lukis. Tapi bila nak menjawab soalan, when you want to answer question, they notice they don't know how to write. They don't know how to explain why they get S, why they get R. Okay, so today's lesson, I hope that I can help you to try to zero in on the technique of answering this kind of questions. Okay, so the diagram, no problem, P and S. All right, then your P, we already mentioned just now, B is big B, small b for your P. S, now S, because it's also a buttercup comb, you can see S is also buttercup comb, so it must be the same, okay, as your P, that means big B, small b. And the next one, you need to write your, all these words at the side, parent genotype, and don't forget your gamete, all right? You must put the arrows to show the big B, small b, they will separate into different gametes during meiosis because of what? Because of the Mendel's first law. We call it that the alleles will separate. Okay, only one allele can have one, only one gamete can have one allele. Okay, after that, you have to combine them, all right, because random fertilization. So your answer for your offspring, okay, anak ya, genotype anak adalah big B, big B, small b, big B, small b, and small b, small b. So here you write in the phenotype, which is if capital B, because it is the dominant trait. So the buttercup, it must be buttercup. And buttercup also for big B, small b, because the buttercup is a dominant trait. That means you need at least just one dominant allele to get your buttercup comb trait. Okay, so it's also buttercup. So big B, small b is also buttercup. And only this one, strawberry comb, is small b, because this is the recessive trait. You need 2 recessive allele to show the recessive trait. Okay, right, next Anna. Now, this is actually the same question, but at the bottom, you need to put the genotypic ratio and phenotypic ratio. The earlier slide, no place to put. So that's why I'm continuing the question here. This is 3 marks. So 1 of big B, 2 big B, small b, and 1 small b. But in phenotypic ratio, you notice that the big B, and big B, small b actually are the same genotype. Okay, that means this one, here they are the same. This one, it shows buttercup comb, so it is 3 buttercup comb ratio to 1 strawberry comb. Okay, now the marks here is 3 marks, but actually every stage here is able to give you a mark. So I do not know how the mark is given for this paper, for this question, but here if you write everything, if this question requires 6 marks or 7 marks, you have gotten the full mark already. Okay, but make sure you do not forget this. I notice a lot of students, they are so lazy, they never write the explanation on the left-hand side. You need to explain what they are. Okay, because I did hear from an examiner, I mean the person who marks the paper, that if you do not write your this one, parent genotype, gamete, okay, offspring genotype, offspring phenotype, all this, you do not get your full mark. You may be penalized also. So please do not forget to write this. I know many students will straight away draw this because they love to draw this, but you have to explain what you're drawing. Okay, all right. So let me go to the next question. Carry on the same question. Actually, it's the second part. All right. Did well. Wow. Exportator. Very good. A minus. Okay. You, after following lessons and following all this, and you study hard, I'm very sure you're going to get A plus for this. If you already get A minus for your trial, that means your A plus is not far away for your SPM. Okay, so keep on working at it, ex potato. All right, okay, let's carry on. How traits among offspring in B8B1 can be produced? Usually this kind of questions come in question 8, That's the last structure question before your essay. So this is one of the difficult, the longer structure questions. Okay, so you say 8B1 is just now lah. Why do you get this? That means the question asks you, how do you get this? Okay, how do you get this trait? So this is where the difficulty comes. Many students do not know how to answer. Okay, you need to answer by combining. You must mention that you get this allele from who? You get this from the father, you get this from the mother, and when you combine, you get your big B, small b, or small b, or whatever. Okay, let's see the answer. All right, 2 marks only. 2 marks only means you don't need to write much. Okay, you just write any 2 points that are relevant, that can explain how you get these traits. Okay, marks. P1, first point. You need to mention that allele B is dominant to, big B is dominant to the small b. Okay, of course, this is already given the question. It may not be very important here. Okay, so you can write other points to support it. You can write this because we do not know what the scheme is until the scheme is released to the markers on marking day, right? On the day that we start to meet for marking. So in order to prepare yourself, please write more because you do not know which of your point will be in the scheme. Okay, jadi anda disarankan tulis lebih banyak daripada yang diperlukan. Jangan hanya nampak 2 markah, tulis 2 markah saja. Don't just write 2 points for your 2 marks. You write 3 points, 4 points, because you do not know whether your first 2 points will get your marks. You do not know, we do not know the scheme. All right, we only know when we start marking. We will not have first-hand knowledge of how the scheme is because we are not in the up there. All right, okay. Secondly, the capital B, the allele capital B is dominant, so it can suppress the effect of the recessive allele B. Okay, we know the effect of dominant is you just need one of the dominant, it will cover or it will suppress the allele of the recessive allele. So the offspring with genotype capital B or capital B small b will show the dominant trait, which is the buttercup comb. So either these two, big B, small b, will show the dominant trait already. Doesn't have to be 2. It has to be at least 1 dominant. It will show the dominant trait, which is the buttercup. And then only offspring with the genotype BB, which is small b, will show the recessive trait, which is the strawberry comb. So in that way, you have already explained why you will get, big B, small b, and so on. So why this one is strawberry comb, whereas the other 3 are dominant trait, buttercup comb. All right, you write in words. So you only 2 marks here. So all this probably you just need only 2 points to be given your 2 marks. All right. Okay, let me carry on. Any 2, all right? Okay, next. Now explain how chicken with strawberry comb is obtained. If both of the parents have buttercup comb, it's possible. Okay, you can get a recessive trait if both parents have dominant trait, but with one condition. One condition is both the parents must be heterozygous. Okay, that means both of them must be capital B, small b. Capital B, small b, and then the 2 small b's there will ultimately join together. You will get 2 small b in the offspring. Okay, I'll show you what I mean. So let me draw this. If let's say your parent here is 1 capital B, 1 small b, all right, both parents must have this small b because you want the baby or the offspring to have this. The offspring, one must get from the father, one must get from the mother. That means your small b must come from one from the father, one from the mother. That means your parents, first point is, both their parents must have capital B, small b genotype. Okay, then only you can get the child or the offspring with small b, because one has to come here, one has to come here. Okay, so both of them produce gamete which carry recessive allele. So you see, because they have a small b here, their allele, their gamete, at least 50% of their gamete will get small b. Okay, you have to have that. both the parent must have at least the recessive allele. Then only the child can have small b, small b. All right, next. When gamete which carries small b combines with another gamete which carries small b, so you see lah, this is your offspring, right? So this one has to combine with this, and then you can get a small b, small b. Offspring with small b genotype is produced. And that is your strawberry cone, okay, which is recessive trait. All right, okay? So it's not difficult as long as you need to know what you need to say. You have to talk about what the parent has, what the other parent has. Then only you can explain what is the trait in the child. So you got to trace back. You know your child is like this, right? The offspring. So you got to know that one of it must come from the parent. So this one come from the mother, this one must come from the father. Therefore, your father and mother must have the small b and also small b. Okay? All right. Next question. Do I see any questions so far? What's the tip? Oh, okay, Vishnu. I think you have not studied yet, is it? Or maybe are you in Form 4? Vishnu? Big B, small b. Oh, big B, big B is we call homozygous, just that the two alleles are dominant. Then big B, small b is we have one dominant allele and one recessive allele. Now, in an organism, you are not able to see it. I mean, you cannot see the difference. You cannot see the difference. Let's say I am big B or big B, small b. Let's say I have, straight hair, right? Let's say straight hair. I will not know whether I'm two dominant alleles or one dominant and one recessive. I wouldn't know. But you can trace it if you look at your family pedigree. That means you see from your father's generation and your grandfather generation. And if many generations are all also having straight hair, all right, then probably you might be homozygous. Probably. Okay. You have to see from there lah, because there's no way you can go and check. There's no DNA test that you can check. You can take up your cell and then you can check whether you are dominant allele or dominant recessive allele. No, you can't do that. Because we cannot check it that way. But how we can find out, like I mentioned, you got to see your family history, whether your father, your grandfather, your great-great-grandfather, you got to see, is there anyone with curly hair? So if probably nobody has been having curly hair for many generations, probably you are homozygous already. Okay, that is how you know. That's how. And there's no way you can check it. Any DNA test also can find it. Okay, so now let's go to question 2. All right, color blindness. So another thing they can ask you is color blindness. Just now was chicken, now it's back to human. Okay, color blindness is a recessive trait. So they say that it's a recessive trait inherited through the X chromosome. It's the sex chromosome. So in one family, the mother is a carrier. So these are the key words that you need to remember. Mother is carrier, father has normal vision. Okay, so complete the schematic diagram below to show how the inheritance of color blindness traits occurs in the family. So they already tell you that mother is carrier. So definitely a carrier has to have one dominant and one recessive of the allele. Okay. So, and then the father is normal. Father is always, okay, of course, male, XY. This allele of color blindness is only found on the X chromosome. So it has to be capital B because he's normal. For the mother is, of course, female is XX. XX is always a female. And then because carrier, she will have one dominant and one recessive. So capital B and small b. Okay, all right. Okay, so let me see. I think there's a comment here. I just see. Oh, thank you. All right, Vishnu, welcome. Right, so you understand, we wouldn't know. We wouldn't know whether I have straight hair, but I wouldn't know whether I'm dominant recessive. I can only trace through my history only. Okay, family history. Okay, so here they already give you the father, all right, the mother. So I know very students love to do this. They just fill up the boxes, very simple. Based on the this is question based on the answer in A1. A1 is actually to fill up first. What is the percentage of the children who are colorblind? Berapakah anak yang buta warna? Okay, now fill up first. Okay, fill up, very simple. Just separate one. This one goes here, this one goes there. Very simple, right? And then you just combine. Now make sure you combine it properly. Afterwards, I'm going to tell you how to combine. Cannot just simply draw lines here. I know many students that simply draw. You get it wrong, okay? There's a way for you to combine. Afterwards, I'll show you in one of the slides. So you get these 4 types or 4 genotypes here, and you have to infer what is the meaning by saying what's a phenotype. So normal, and colorblind. Okay, because capital B is normal. Capital B, small b is also normal, but actually she's a carrier. Capital B, small b is normal but carrier. Okay, this one is definitely normal lah because there's no recessive allele, capital dominant only, and this is colorblind. So now answering this question, how many percentage of the children are colorblind? Okay, so if how many percentage of children are colorblind, look at how many children. Okay, tengok ada berapa anak. You're looking at anak, children. So you see 1, 2, 3, 4. Right, total 4. And how many are colorblind? Only 1. 1 out of 4. 1 out of 4, because of percentage, you have to times 100%. 1 out of 4 times 100% is 25%. Okay, so even though it's carrier, we also consider as normal. All right, now, what if I change the question? What if I change the question to, based on the answer A1, all right, what is the percentage of boys who are colorblind? Look carefully. They're asking about boys now. Okay, what is the answer for this? Maybe some of you are here with me today. Maybe you can try to answer. Okay, so what is the answer for this? What's the percentage of boys who are colorblind? Okay, let me just double check. Any answer? It's not difficult. It's not difficult. You just need to look at the boys. Yes, you mean correct. Wayjet, Brandon, correct. 50% now. Okay, why 50%? Because we are looking at boys only. And here, there are only 2 boys. Okay, XBY and XB capital Y and X small b y. So no need to look at the girls already because it asks for boys. So it's 1 out of 2. 1 over 2, your answer is 50%. Okay, so be careful before you answer. Look carefully. Are they looking for children? If it's children, you include the girls as well. Okay, over 4. If it's boys, you only look at the boys, which is over 2. All right, okay, I think you all got it right already. Okay, very good. Okay, I've got excellent students here. Okay, you're going to do perfectly well for your biology later. All right, now, an O-blooded man marries an AB-blooded woman. What is the possibility that the child will get blood O? Explain why. Okay, now what's the possibility first? So you need to know that if the father is O-blood, what kind of alleles will the father have? And then AB blood, all right, the woman has what kind of alleles? So remember, we have 3 types of alleles for blood grouping: IA, IB, IO. There are 3 types of alleles. And then what kind of phenotype the person has is a combination of any 2 of these. Okay, let me see the answer first. What is the possibility? Any possibility? Okay, I think your 50% was for the previous question. Chi Ming Hui, Zina, correct, all 50%. So what about this question? Is it any possibility? If there is, what is the percentage? If there isn't, that means 0 lah. All right, if there's no possibility, you put 0%. Okay, any percent? 0. Wayjet says 0. You mean 0. X potato 0. Okay, very good. 0. Okay, why? 2 marks here. No possibility. Tiada kemungkinan. Okay, because let's try to draw the schematic diagram. If it's an father is O, O blood, the mother is AB. In order for the father to have O blood, it must be IO. There's no other possibility to get O blood. He must have 2 recessive alleles with O here. For the mother, it has to have IAIB. All All right. And then if you try to draw a schematic diagram of what kind of children they get, you will notice that this is IO, this is IO. Okay, just to simplify it, if there's only this, both of them are the same, you can simplify by just drawing one. Okay, you can just draw one. And then when you want to combine, this is IA, 50% of it, of the gamete will have IA, 50% will be IO. So you just combine this one, you get IA, IO. This is IBIO. That means all their children will be either IA or IO. That means A blood or B blood phenotype. It's not possible to have a O baby. Okay, then you know the paternity issue and all that. If your father is O, mother is AB, then if the child is O, you know what happens, this child is not his child. Okay, you understand what I mean? This is some of the ways that you can find out. All right. So, what is the reason? The mother does not carry any IO. In order to have a baby with O genotype, phenotype, one must come from the father, no problem. Father already got IO, but the mother does not have IO. All right. In order to have an O, the baby must have IO. So the one come from the father, then this one must come from the mother, but the mother doesn't have IO. So that is the reason why. Okay. If you want to explain further, you can explain like this. Okay. In order to produce an offspring with O blood phenotype, there must be 2 IO alleles from each of the parent. That further explains it. Okay, so but these 2 is enough to get 2 marks. But you want to explain more, all right? You're afraid you do not get enough answer. Do not get your answer will not get the marks. You can be more explicit. All right, you can explain more. In order to produce an offspring with O blood phenotype, there must be 2 IO alleles, and they must come from each of the parent. Father must have one, mother must have the other one. Then only can get IO. Okay, fine. Okay. Any question? Do we get marks for drawing? Because only 2 marks only, they want the explanation. Because if you want the drawing, they will ask you. If there's marks for the drawing, they say draw a schematic diagram. That means you definitely cannot escape. You cannot substitute your answer by writing in words. You can get some marks, but you will never get full marks. If they say draw a schematic diagram, make sure you draw. Okay, and also put in some explanation in case to cover up something that you missed out in the diagram. But like in this case, only 2 marks. Okay, you need 2 marks only. It need more of explanation. Why? You don't need to draw for this. You can draw on the side as your own working to understand what you need to say, right? What you try to remember back how to get a child IO. You must IO come from father or no? IO from mother. You can draw it at the side. But in this scheme, I believe the mark is not for drawing. You have to write this point, the F point and the P point. Okay, this is only 2 marks. That's why they want the explanation. All right, Rayjet, I hope it's clear to you. Okay, let me carry on. Pedigree diagrams is to show the family, the grandfather, marry this one, male marry the female, what children and so on. That is like a family tree. That's called a pedigree diagram. This is inheritance of a royal family. This is a real case, Okay, so the what they call the UK family, the Queen Elizabeth and the family, all these are actually they are a little bit related to the cousins and all that, the royal family in Germany, the royal family in Netherlands, the royal family in Russia and all that. They were actually related because they have actually this hemophilia gene in their generations. But fortunately for England, UK, that already did not, Elizabeth did not inherit that. So Elizabeth was not a carrier. Elizabeth, this queen, the Queen Elizabeth, that queen is not a carrier. So that's why in the further generation, the Charles and then all the children and the grandchildren, Prince Harry, Prince William, all don't have, because Elizabeth was not a carrier, but her cousin was carrier. No, don't have. Okay, so it started with Victoria. Queen Victoria was a carrier. And then, kerabat diraja, they always marry among always their family, they marry the royal family. So this royal family of this country marry another royal family of another country. So they keep it through the the royal family. So that's why now, later on you can see certain royal families, they will still have the hemophilia case there. All right, but for the UK one, no, because Queen Elizabeth didn't happen to be a carrier. Okay, now 3 marks. What is the question first? Why are boys more likely to get sex-linked diseases? This is a very common question. Why are color blindness and hemophilia, these are two sex-linked diseases, why are they more commonly found in males? Okay, so it has to do with the sex chromosome X. Okay, this allele that causes this disease, whether color blindness or hemophilia, this allele that causes a recessive allele is only found in the X chromosome. Okay, now, so here's the question again. All right, so here 3 marks. How to answer? All right, you know, many of you know, but don't know how to say it, don't know how to write it. Okay, now, so first of all, the Y chromosome for the male, in order to get a male, you must have a Y chromosome and an X chromosome. So the Y chromosome does not carry the gene that causes hemophilia. Okay, the gene that causes hemophilia is found on the X chromosome. So for the man or the male, you only need one X and one Y. So if your X already got problem, that means your X has the recessive allele. You just need one only, and it will show the color blindness. Okay, let's say you already have color blind. You don't need two. For the female, on the other hand, if one is dominant and one is recessive, the person is still normal. Okay, so it's like less chance for a girl to show this sex-linked diseases, because you still have another chance. You may have a dominant for one of it, the other recessive, never mind, because the dominant can cover, menutup, or we call it can suppress the effect. But for boys, no chance. You just need one only. Whether you're so unlucky to get the allele, then there you are. You already have colorblind, or you are already have hemophilia disease. Okay, so second point, males only have one X chromosome. That's what I mean. Males only have one. You do not need two in order to X chromosome to form the male. All right, you only have one. So the recessive allele, a hemophilia allele on X chromosome in males can express itself. So if you only have one, it already will show. It can express itself. That means it shows the trait. It shows the color blindness or hemophilia. Okay, and the Y chromosome cannot suppress, because the Y chromosome, the locus, I mean, this dominant allele is not found on the X chromosome. It's not found on, it's not found on the Y chromosome. So your Y chromosome cannot cover the effect. Cannot cover the effect of hemophilia carried by the X chromosome. So for boys, too bad you just have one chance only whether your X chromosome is the normal one or is the one with the recessive allele. So if you get that recessive, that's it. You don't have another X chromosome to cover the effect. Okay, so these are the points that is needed. All right, so that you can explain it in that way All right. Okay. Next, so this is something similar. This is hemophilia from the generation 1 to 3. That means the father generation, then go on to the children and the grandchildren. Okay, so look at the coloring here. Be careful. White means normal, and then the shaded one in squares is a carrier. Then the one that is darkened or blackened, that one is hemophilia. Okay, so 3 marks. What is the question? Based on diagram 8.1, state the genotypes of father and mother. Okay, so very simple. Father is, you see, white. White means totally normal, right? And for the mother is the carrier. Carrier, you always have one dominant, one recessive. All right, so the answer here will be hemophilia. So we put H. For mother is one dominant, one recessive, capital H, small h. For the father is capital H only. Never put it on the Y, because your allele for this disease is not on the Y, always on the X. Okay, the father is normal, so capital lah. Okay, next. All right, next one. J. The generation 2, that means the child. All right, from this father mother, you get the child is J. J is a daughter, got married with a man. All right, the man. So it is found that their children inherited hemophilia. So got hemophilia. So if there's hemophilia, that means the recessive gene must be in the parents already. Okay, now complete the schematic diagram to show the inheritance of the disease. So we just now we say what it was J. J was now J is this one. See, J is a carrier. So the girl is a carrier. That means the mother is a carrier. So the mother is a carrier. You have to put like this, capital H, small h, X capital H, X small h. Then the father must be normal. Okay, the father is normal. So you will get the next generation, the next the gamete will be capital H, Y, and then you have the generation one, that means the children. You have capital normal. Then you have girl normal, girl carrier, boy normal, boy color. No, hemophilia. No, hemophilia. Okay, so here 3 marks. Usually you will have these 3 levels to give you the 3 marks. If you have 6 marks, that means you must see the line also. They will give you marks at the line. And also don't forget your phenotypic ratio and your genotypic ratio. They will also have marks there. So always be careful, write full. Don't care how many marks it is. All right, you write the whole diagram full. Never leave out anything. And of course, don't forget this one. This one is very important. All right, next one. Now, hemophilic patients will face problem in case of injury. All right, that means there's any injury, cut, or that you start to bleed. His blood could not clot because there is no clotting factor in the patient's blood. Suggest a genetic technology. This one tie in with the chapter 13, which is genetic engineering. How to help this patient. Okay, so no clotting factor. So we will need to produce that clotting factor. Usually we make use of bacteria to produce it because bacteria can multiply very fast. Their reproductive rate, their rate of having the next generation, all that is very fast. So in that way, it can produce a lot of the substance that you need. That's why we also make use of bacteria to make insulin. Okay, so the same principle of making insulin. So you need to carry out genetic engineering to produce transgenic bacteria. Transgenic bacteria is bacteria that you have already changed the DNA. That means you put the ability or the DNA to make the clotting factor, you put inside the bacteria. Okay, so this bacteria is called transgenic bacteria or bacteria with recombinant plasmid. That means the plasmid is the DNA in the bacteria. You already put the DNA for making clotting factor inside. Okay, so it makes the bacteria able to make the clotting factor now. Can make already. Okay, so 1 mark only. So you need 1, or you can have another answer. Carry out gene therapy using modified virus. So you have a modified virus, normally adenovirus, when you put the DNA inside and you let the virus inject this DNA into the cell of the person. That is called gene therapy. Using genes, you insert the gene into the body of the person using virus. Okay, and that the person will be able to produce the clotting factor because your DNA to produce the clotting factor already put inside. Okay, all right. Okay, so 1 mark on it. So it doesn't need a lot of explanation. Just need to know the keywords and the correct words to use. Use the terms there. Okay. Next one, anyone. All right. Now, question 3. State the type of variation. Now, this is the same thing as the hemophilia disease. So hemophilia is very, very straightforward. Either you have or you don't have. So what kind of variation is this? When you have or you don't have. So it's nothing in between. Okay, so this is called continuous variation ataupun variasi tak selanjar. Sebab dia tak ada ciri-ciri perantaraan. Sama ada you ada atau you tak ada. Tak ada di tengah-tengah. Okay. And then explain the cause of this type of variation. So why do you get this type of discontinuous variation? Okay, it's because of the genetic mutation, gene mutation. Right, you get the hemophilic problem because the gene is abnormal, caused by gene mutation. Disebabkan oleh mutasi gene. The gene that controls the production of clotting factor has undergone permanent changes. So the gene that controls making the clotting factor, this gene is already changed. So when it's changed already, it cannot make the clotting factor. So that means the cell, the person, the body cannot make clotting factor. Okay, so the gene changes causes the clotting factor not to be produced. So you need to have the F plus any one of these points. Okay, understand? So you need to say that it cannot produce the clotting factor because the gene that controls it is already been changed because of mutation, right? Okay, next. Now this is an essay question. Okay, I just intersperse with the essay question. Now this is a genetic disorder. Okay, so frequent and overexposure to ultraviolet radiation, UV light, can lead to mutation. Okay, this is the Rhesus mutation, UV light. Down syndrome and hemophilia examples of genetic diseases caused by mutation. Okay, so we're talking about mutation now. Okay, so how is hemophilia inherited? How do you get hemophilia? So you got to look at the problem. I mean, what causes it? And like I mentioned just now, hemophilia is caused by a recessive allele, all right, which is found on the X chromosome. So it makes the person unable to produce the clotting factor. Okay, so hemophilia is inherited by a recessive allele. Okay, this recessive allele, and it is found on the X sex chromosome. All right, it's a simple one, just simple one, 2 marks only. If you are afraid you do not get the full marks, you can write further information. Like you said that this recessive allele does not have the ability to produce the clotting factor. You can write more, no harm. Essays, please do write more. You are very encouraged to write more. Of course, not too much until you got no time to answer the question. All right, you can add in a few more points just in case your point that you write earlier is not in the scheme. Okay, next. State the risk faced by hemophilia. Hemophilia is a person who has hemophilia. Explain how this affects her quality of life. Of course, definitely no good when you are prone to injury. Whenever you have a skin cut, you know that you're going to bleed and you can potentially die from the bleeding. So 2 marks only, again, very, very little, right? Don't be worried. Just answer little question like that. You can get a lot of marks from that. Extensive blood loss. When the person gets injured, definitely the person will bleed uncontrollably. All right. And how to solve this problem actually? All right. This person has to be hospitalized. You have to bring them to the hospital. They have to have injection of clotting factor. So the doctor there will inject clotting factors to help the blood to clot for the injury. Okay, but it's not going to be permanent. Every time the person is going to get injured, you have to bring the person to the hospital and they have to be injected with the clotting factor. Okay, next. Of course, the patient will become weak and lethargic. Lethargic means tired, lemah dan lesu. Okay, because one of the reasons is because you are losing blood. You're losing blood, that means you're losing red blood cells. And you know red blood cells carry oxygen. So when you're losing red blood cells, less oxygen will be carried to your body cells. So you have lack of oxygen, you will feel tired, you will feel weak. So these people usually do not have a very active lifestyle. They cannot run, they cannot do strenuous activity in case they injure themselves. All right. So they are also very tired and lethargic most of the time. Okay, can be life-threatening. Boleh mengakibatkan maut, means the person can die. So of course, the quality of life is very, very low, I would say. They're always in potential danger. Okay, next. Essay, B part. State 4 differences between hemophilia and Down syndrome based on this criteria. So when I say based on this criteria, you must mention it. You must, because marks are given for that. Any additional points you can add in, but do not skip this 4. Okay, so what causes it? What is the number of chromosomes in these patients? What is the characteristic? Talk about the symptom. What will the person show? And how is it inherited? Okay, so 4 marks. Okay, 1 for each one. So D5, I give an extra point there. So first thing is caused by non-disjunction of the chromosome number 21. That means during meiosis, all right, somehow the pairing of the chromosomes, they did not split evenly. So 2 of them went to one side, whether it is the homologous chromosome, okay, or the sister chromatids, right, during anaphase 2. Okay, then hemophilia is caused by recessive allele on the sex chromosome X. Okay, recessive allele. So just basically write down what is the cause. Caused by what? Okay. Second one, your answer about the number of chromosomes. Down syndrome will have 47 chromosomes. Hemophilia, they have normal, 46. Okay, it's nothing to do with the number that is abnormal. It's just that the allele is abnormal, not the number. Okay, then what about the symptoms? What was a Down person will show? What kind of symptoms? You have a flat face, White face, okay, slanted eyes, mata sepet, and then the protruding tongue. Sometimes they can't close their mouth because the tongue is a little bit big. So that's why protruding tongue. Sometimes they will salivate, right? You can see saliva coming out and all that. Mental disability a bit lower lah. That means the IQ is a bit lower, right? Not the normal person IQ. Then this hemophilia, just 1 point only, just the blood unable to clot under normal situation lah. Okay, then next one, how is it inherited? For Down syndrome, it's not inherited from parent to child. So if normally a Down parent, it's not for sure that you will have a Down baby. Okay. For hemophilia, this one is inherited because of the allele that is found on the X chromosome. So that one definitely is inherited. All right. Okay. And the extra point here is Down syndrome is an example of chromosomal mutation where the number is abnormal, the chromosome. For hemophilia, this is an example of a gene mutation. Okay, not the chromosome mutation. It's a gene mutation. Okay, next one. Now, this is on the Rhesus factor. So they tie in with the blood group also. So D antigen found, there's a factor called Rhesus factor found on the surface of red blood cells. Individual red blood cells that contain Rhesus factor are referred as Rhesus positive. So you can either have Rhesus, you are Rhesus positive means you have the antigen. If you have the D antigen on the surface, like this one, then you are considered positive Rhesus. If you don't have that D antigen on your red blood cells, you are considered Rh negative. Okay? The positive is dominant, the negative is recessive. All right? Now, a husband has heterozygous dominant Rhesus positive blood. So the husband, father is heterozygous dominant, heterozygous dominant, that means one is dominant, one is recessive. Then the wife is a Rhesus negative. In order to be Rhesus negative, you must have two recessive negative. Okay, using a hybrid schematic diagram. Now here, like I said, this one you must draw lah. Widget. This one you cannot skip already. Using a hybrid schematic diagram, explain the probability that the children have a Rhesus positive blood group. Okay, so here, how many percent? What's the probability the child has a Rhesus positive? Okay, so here, drawing again, same thing. Start with the phenotype first. Father is this, mother is this. Father is hetero because given in the question. So if hetero, mother is negative, must be negative, negative. That's the only way to get Rhesus negative. So next one, I'm going to tell you how to draw the fertilization line carefully. So you just split into two, you get your gamete line. After that, now here, fertilization, do not draw randomly. I just want to revise. There's one very good method which I always tell my students to draw. You draw one line from the first gamete. So label your gamete. This is considered first gamete, second gamete, third gamete, fourth gamete. You combine your first and the third. First and third, 1, 3. Okay, and then you just immediately write under that. Do not join all the lines first and then you write because you will get mixed up. When you see the lines, you will get all your mata will become pening already. You will draw wrongly. So do one first, write Rh positive, Rh negative. That's one. One done already. Now the second one, you must take the one again and then you combine with the one that has not been combined. 4, right? 1 and 4. All right. Now do not do 1 and 2, because you you cannot combine a sperm with a sperm, right? You must take one sperm with one ovum. So take one sperm, one ovum. So 1, 3 is the first one. Then go for the next one, the ovum, 1, 4. Okay, so it's again positive, negative. Now then you must also use the second sperm there. I mean the Rh negative, the gamete 2. So you take your gamete 2, and you combine with one, the first one from there, the female, which is 2, 3. 2, 3, you get Rh negative, and don't forget the other combination, 2, 4. Okay, 2, 4, you get also negative, negative. So remember this keyword, the number, the pattern, 1, 3, 1, 4. 2, 3, 2, 4. You will never go wrong. Okay, some people, they draw the lines very nicely, it's like very symmetrical. Actually, it's wrong because they repeated 1, 3, and then again 1, 3, 2, 4, and again 2, 4. It looks nice, but it's wrong. Okay, so remember 1, 3, 1, 4, 2, 3, 2, 4. Okay, next one. You get, don't forget your phenotype, then the ratio 1 positive, 1 negative. Okay, always your ratio must be the smallest number possible. Okay, so here, where are the marks coming from? If it's 6 marks, every line will have a mark. Okay, usually 6 marks. They have 7 here, they will give you maximum 6 already. So that's why always when you draw the full diagram, because you will never know where they give you the marks. Okay, next, variation is differences among organisms of the same species. All right, now you can see cats here. The mother here has a different fur from the babies. All right, you can see this is variation. The mother doesn't look like the baby doesn't look exactly like the mother, because there's variation from the parent to the child. Okay, so 6 marks. See, this is a little bit more. So look, how does this situation occur? That means, how do you get variation? Okay, how do you get variation? How come the child doesn't look exactly like the mother, even though they come from the mother and father? Okay, so you have to look at the marks, 6 marks. Make sure you got 6 points to write. Okay, crossing over. Now, because when you talk about variation, you can never forget about crossing over. You have to say crossing over atau pindah silang. Start by saying pindah silang berlaku. Even if pindah silang berlaku, quite likely you get 1 mark. All right, even though you didn't say the detail between what and what Don't worry about the detail. You must say pindah silang. Okay, pindah silang or crossing over occurs between non-sister chromatids of the homologous chromosome during the prophase. So you got 2 X's here. These are the homologs. They are together, and then one part of this, they will overlap. They are non-sister. Non-sister means if it itself is sister, they will never crossing over between sister chromatid. It is always non-sister chromatid. Okay? That's during phase 1, prophase 1. Then this produces new combinations of gene, menghasilkan kombinasi gene yang baharu. Okay? We call recombination or recombinant. And then again, this is crossing over is one of the causes. Another cause of variation is because of how they are arranged during metaphase 1. How the chromosomes after crossing over, they are arranged. They can arrange like this. They can also arrange like this. If you arrange like this, you have another possibility already. So you see now, so the second feature or the second factor is actually random arrangement of homologous chromosome during metaphase 1. It can be like this, it can be like this, because they are able to move around when they are, they can susun, we call it arrange itself. Okay, next. So, and also after that, the chromatids will separate during anaphase 2. All right, and also they will produce different gamete with different genetic composition. All right, and then the third factor is because of random fertilization. So remember, there are actually 3 factors, including the 4th one is mutation. Variation is caused by number 1, crossing over. Number 2, random arrangement of homologous chromosome during metaphase 1. The third one is random fertilization of gametes. So there are millions of gametes, okay, for the sperm. And also, of course, there's only 1 ovum, but doesn't matter which one gets to fertilize, that is also a chance by itself. And then lastly, okay, number 7 is referring to the 6. So random fertilization of gametes produces zygotes that have different genetic information from its parent. So you see the keyword here. You must write the word different. Different. Berbezer. That's why it's called variational. You will have berbezer. Different genetic information from the parent. And lastly, don't forget about mutation also. Mutation also gives you another avenue for producing variation. So you can be either gene mutation or chromosomal mutation. You can just say mutation can produce individuals with different traits. So you mentioned 4 already. 4 factors which causes variation. Okay, revise this because a lot of students usually cannot answer. They cannot explain this. All right, they cannot really see how it works. But doesn't matter if you can write all these things. Definitely you will score. You will get your 6 marks. Here are 8 points. You will definitely get full point already. Okay, just every time they ask about variation, how it works. Okay, write all these 8 points. This is perfect. Okay. Next, any 6 points. Next one, question 5. Okay, so this is on pea plant. All right, pea plant. So this is a simple all right. You've already seen this is dihybrid crossing. All right, inheritance. So okay, B, capital B is for bulat, round, and then small is for wrinkled, right? And then capital G is for yellow, small g is for green. Okay, so here, all the offspring are round and yellow. So you see here, all of them are round and yellow. That means this must be purebred. This is a purebred dominant trait. This purebred recessive trait. In order to get all of them heterozygous, okay? Blue, I mean round and yellow. So it must be this one heterozygous. Okay, so now one of the anak, okay, so one of them, now this is a P, which is actually supposed to be a big B, small b, big G, small b, is going to be crossed with a Q plant with this genotype. So first of all, you'll fill in first. Fill in this one. Big B, small b, capital G, small g. Okay, and then they already give you the gamete, all right? The gamete. Remember, take one of the B, take one of the G, put in together. One of the B, and don't repeat, big B, big G, big B, small g, small b, big G, small g, small b. All these are different combinations, but you must have one for the seed shape, one for the seed color. Okay, next. Then you just combine. Now this one, capital B, capital small G, there's only one. It only produces capital B and small g. So you can just write one only. And later you combine everything, right? You're going to get this. All right, so you just complete the schematic diagram. All right, next, R. R is referring to this one, R. So now the next question refers to R. Capital B, big G, small g. Explain how offspring R inherits round and yellow seeds traits. So it has dominant for round, but it's heterozygous for seed color. How? Again, students don't know how to answer, right? Don't know how to write. So don't worry, we must say get what from the parent. R inherits two dominant alleles because remember this is the R, capital G, small g. Okay, this is the R. So you get two dominant alleles of seed shape from its parents. So one from the father, one from the mother. Okay, and then what about the G? R inherits one dominant from the father, G, and one recessive allele for seed color. Okay, so you must say get what from the father, get what from the mother. That is the way to answer these questions. Then the R inherits at least one dominant allele for seed shape. So at least one dominant. So this one, two dominant, never mind. It will still be dominant trait. But at least must have one dominant. And then for the seed shape, and at least one dominant for the seed color. That's how you can get brown, which is a dominant trait, and yellow is also a dominant trait. It's a dominant trait also. Okay, so remember, say this one get what from the father. This one get what from the mother. Say like that. Okay, next one. So this is example. Now this is for purple flower, white flower, purple flower. This is monohybrid. Just now the seed color, because there are two things, the seed color and the seed shape. That one is dihybrid. So now the question asks you to state the differences. That means they're asking you to state the differences between dihybrid and monohybrid crossing. Okay, just this is monohybrid. This is mono. Just now earlier one was dihybrid. Okay, so what are they? How to say the differences? Very simple. You can mention the inheritance because it's not given the name. So whenever the name is not given, it could be a mark that is given there. So you say that diagram 8.1 shows dihybrid inheritance, while diagram 8.3 shows monohybrid inheritance. Okay, then describe the difference between the two. For dihybrid involves two characteristics of the plant. So that means the seed color and the seed shape. For this one, it's just the flower color only. It involves one characteristic. When you talk about the color, no need to think about whether the plant is tall or short, whether the flower is axial or terminal, no need. Okay, next one. It involves two pairs of alleles when it's dihybrid. When this one involves one pair of alleles. Okay, mono. And for dihybrid, you will get 16 genotypic combinations. When you draw the Punnett square, remember the Punnett square? You will have 16 boxes for your Punnett square. But for monohybrid inheritance, you will only have 4. All right, 4 genotypic combinations. So it's less complicated, definitely, monohybrid. Then you can have another point here. For dihybrid inheritance, it will involve the 2 Mendel's law. Mendel's first law and second law of independence. Okay, law of inheritance, not independence. Sorry, first that Mendel's first and second law of inheritance. Wrong. Inheritance. And this one involves, for monohybrid, it only involves Mendel's first law of inheritance. Okay, sorry for the typo. Too many words to type. Sometimes you get confused. Okay, so any 3 pairs, you get 3 marks full. Okay, not a problem. You need to know the difference between the 2 types of crossing. Okay, we're coming to the end. There's one more question, question 6. So this is the phenotype and the genotype of plant P plant S. So you have purple flower because you have at least 1 dominant is purple. Short tree, 2 recessive. So that means capital R is tall. That means the small one, small is short. Okay. So plant P plant S is crossed with P plant T. So this is S. This is S. Identify the genotype of P plant T so that the cross will produce all offspring with phenotype of purple flower. How is it possible to get all offspring with purple flower and short tree? This one is a little bit okay, but you got to work backwards. You need to know what you want to get. So I'm going to rub this away. My working is here. Okay, example. So you need to know that your offspring, if it has to have purple flower, it must have at least 1 P, capital P. And in order for it to be short tree, it must have 2 small r. So the offspring must have, could be either capital P, small r, or this one pun boleh. So the offspring must have this type only. Cannot have small p, big R, cannot. So your offspring, you must find the other partner, which is your T. What is the genotype? So S times T. In order to get your only this anak you have. You cannot have other combinations. So you got to work a little bit. Maybe you need a piece of paper. You got to draw, and find out. Okay, so I show you working. You know that S is this big P, small r. But you do not know what is your T, right? You do not know what is your T. All right, never mind. You know that your S can produce this kind of gamete. Big P, small r. Small p, small r. All right. And then you don't know about these 2. All right, never mind. Leave it first. Then you want to know, you need to know, you already know that your offspring must be these 2 type only. Must be these 2 type. You cannot have others. That's how all have purple flower and short tree. Okay, now, then this one must be what? In order to get this, you work backwards. All right, you have your capital P here. Sorry. Your capital P here. It's not working. Okay, here, I'll show you here. The genotype for P plant must be capital P, small r. Okay, must be. You know why? Because the offspring must inherit 1 dominant allele for purple flower from both. See? Here must be capital. So here also must have a capital. Here also must have a capital. Here also must have capital. So if both have a capital, that means this one must be capital P already. Okay? If this one is capital P, this one capital P, this one must be. You cannot have a small p here. Why you cannot have a small p here? If you have a small p here, this can combine with this, you're going to have small p, which is not possible because the question said all must have purple flower. So this one is going to be white flower. So cannot have a small p here. So this means this must be a capital P. So when this is capital P, you go backwards and see, you go and join backwards, you must have a capital P for the parent. Okay, now look at the small r block. The offspring must inherit 2 recessive allele for short tree from both parents. So you know that it's small r here. If small r here, the R must come from this father and also must come from this mother. So this one must be a small r. and then you go and join backwards. Your father, the mother must be small r. You see that? Okay, the offspring must inherit 2 recessive allele for short tree from both parents. So because here is already here, small r, one must come from here, one must come from here. So all the gamete must have small r. That means the mother must be this type, not mother lah, the other parent lah. Understand? You have to work backwards. This is what I mean by working backwards. See the anak first. Tengok anak dulu. Lepas tu cuba fikir. Tengok, dia mesti dapat apa? Dia must get what from in the gamete, mesti dapat. Gamete itu mesti membawa apa? The gamete must carry what kind of allele? Okay. So P plant S produces gamete with allele P, capital P R, and another one is capital S. Oh, sorry, this is capital. Okay, this is wrong. I wrote them wrong. This is wrong. Okay, so capital PR, capital P and R. Capital P and small r. All right, so there you have it. You have your parent already. T. P plant S. Sorry, you're referring to PS. Sorry. Yeah, correct. It's referring to S. Yeah, referring to S. All right, okay. So that's how you answer. You have to work backwards. Okay, now let me go on. So the last one is the gametes of P plant T must carry PR. So it must be PR. Only. Capital P, small r. Okay, I know that this is one of the difficult questions when they ask you to look at the possibility of the indok of the parent. Okay, let's go to the last question. This variation, right? 5 different 5 individuals of the same age. This is one type of variation. 8.1 is one type, 8.2 is another type. Okay, I'm sure you know by now. This one does the differences are very slight in terms of height. There is intermediate value between one person and another person. But for this one, it's very obvious there are 2 types only. This is a free this is the attached earlobe. This is the free earlobe. Ada 2 jenis saja. So one is continuous. Now ask for the type. Simple. For 8.1, it is continuous. 8.2 is discontinuous. Okay. Next one. Simple question. State 2 differences. So I have a lot of answers here. Just any 2 will do. Differences. This is a very standard question. So talk about what? The name, because name is not given. We can talk about 8.1 is or shows continuous variation. 8.2 shows discontinuous variation. 8.1 has intermediate characteristics. That means in between. There are difference. The difference is very slight in terms of height. For 8.2 does not have intermediate. Okay, it's either you are this or you are that. You are free earlobe or you are attached earlobe. Next one, differences are not distinct. Okay, for your 8.1, differences are very distinct. This type and that type are very different. Next one, can be measured is the you call it quantitative. For height, you can measure it. It is quantitative. But for the other one, it's not measurable. It's either this or that, the shape only. For 8.1, this is the continuous, influenced by genetic and environmental factors. So it can also be influenced by environmental factors. Okay, that means the person eats good food, you may grow taller, right? Or the person is malnourished, you will not grow that tall also. So it has to do with nutrition as well, which is environmental factor. For your earlobe, there's nothing you can do, nothing you can eat, whatever. It was not going to change your type of earlobe. It's because you're born with the gene. So that's why it's influenced by genetic factors only. All right. Then lastly, you have controlled by more than one gene for your continuous, and for the discontinuous, usually controlled by a single gene. Okay. And one more, I think last question. How does albinism work? How does it happen? Albinism is a genetic disorder that causes melanin pigment cannot be produced or not to be produced. This melanin gives you the brown color in the skin. These pigments fade can block out, not only fade, block out about 99.9% of the UV rays from the sun. Describe how albinism occurs. How do people get albinism? How come suddenly their skin becomes so white? Okay, why they can get a problem with their pigment production? It's because they cannot produce the melanin. Okay, 2 marks only. That means nothing much. No need to write a lot. You say caused by gene mutation. This is definitely an example of gene mutation because the gene cannot produce the melanin, okay, which causes a change in the nucleotide base sequence of a gene. So the gene, all this ACT gene, all that, the order has been changed. So it's no longer the correct gene to produce the protein called melanin that controls the production of melanin pigments that which gives color to the skin, the hair, and the eyes. So that's why people who have albinism, the skin is very fair, very white. The hair is also blonde, white, and the eyes, the eyebrow here, and then even the coloring, the iris is very, very light. So because you don't have the melanin, okay. So two marks only. That's why you don't need to write a lot. Okay, what steps should be taken by albinism? I mean, albino. The person is called albino, not albinism. The English not so good. Albino is a person. If they want to carry out outdoors activity during daytime, this is common sense, right? This is checking your understanding and your kebat lah, right? So you can write any answer as long as you protect the skin from the UV light. So how to protect? 1 mark only. Wear sunglasses, a hat to protect the eyes or the face from the UV light in the sunlight because they will sunburn easily. The UV light can cause cancer because they do not have the melanin to block out the UV light. Okay, so they have to be extra careful when they go out. They have to wear hat and so on. And then another one, you can use umbrella, wear long sleeve shirts, long sleeve pants to protect the skin from from exposure to the UV light in the sun. Okay, this is any person can answer because just common sense. All right, so insulin. Last question. How do you produce insulin? All right, this one is the genetic engineering, chapter 13. Okay, to treat the disease called diabetes mellitus, which is penyakit kencing manis. Okay, there is glucose in the urine. So how to produce insulin? This one using bacteria. Okay, using genetic engineering technology or DNA recombinant technology. Okay, either one you get the F first. F is the fact. You must say what technology it is. Say the technology name. Then you explain the steps. Okay, so human DNA with insulin gene. You must take out the human DNA which produces the insulin. That means must be from the cell in the pancreas. You must isolate the cell in the pancreas and then you must isolate the DNA. You cut it out using restriction enzyme, not scissors. Don't say scissors. Okay, after that, you must take out and cut it with the restriction enzyme. So you have two things now. The human DNA with the ability to produce insulin, and you have the DNA from the bacteria. All right. And now you need to insert the insulin, the DNA, the DNA which the ability to produce insulin, you must put it into the plasmid. Okay, you refer to the diagram in your textbook. It has a very good diagram to show you the process. All right, refer to chapter 13. I'm sure you have a textbook there. All right, so insert inside now. That means this plasmid now carries the gene that can produce insulin. So you need to put it back into bacteria. Okay. The recombinant plasmid, now it's called recombinant because it's no longer the original one. It's put back into the bacteria, and now you let your bacteria multiply. As I mentioned earlier, bacteria can multiply very fast. Okay. So bacteria will clone, the clones will multiply and produces insulin in large quantities. You take out the bacteria, you extract the substance there, you will get insulin. You purify it. Then you put it into bottles, and then you send to hospitals all over the world, right? And then to be distributed to diabetic patients. So insulin is extracted from the bacteria and purified. Okay, so you see, 4 marks here, you only need 6 points here, you only need 4 points. All right, okay. So here, you need to talk about the fact first, which is your F plus 3 points from here. All right. So you see, all right, so I've already explained certain questions. I hope these questions will be able to help you, guide you to answer, explain, especially the schematic diagram that most people do not know how to explain. So you can rewind this, okay? You can watch it again. If you need to copy, just pause it, all right? No need for me to share my slides to you. You just pause it and there is your slide. You can copy at your own leisure. Okay, so harap telah membantu murid-murid semua dapat maklumat yang penting untuk menjawab soalan-soalan jenis ini. Okay. Why do we get marks? Okay, I already answered. Yeah, Reject's answer. Okay, so everyone, I hope to see you again. I'm still going to present some more answering techniques to help you until you take your SPM on the 15th or 16th of March. All right, okay. So please do ask me questions if you need to. All right. My Telegram. Wait, I'll give you my name. Telegram. So it is here. You can just message me if you have any questions. Okay. So this is my Telegram username. All right. Okay. I'll see you next lesson. All right. Maybe next Saturday, or hopefully, if I have enough time to do another slide, another set of slides, I will have it next Saturday. Okay. See you then. All right. Bye bye, everyone. Have a good evening. Have a good weekend, and do study hard for your exam next week. All right. All the best. All the best. Oops cap. What is oops cap? Oh, thank you, teacher. Okay, no problem. All right. Thank you. Bye bye. See you.